Welcome back, students, to the sixth video of our online classes in pharmaceutical microbiology. The last class, we have dealt with the various methods of isolating pure culture. After that, we have seen the growth curve, how the bacteria is growing, and how is it absorbing the nutrients, and how is it declining in four different phases. That was lag phase, log phase, stationary phase, and decline phase. So we have learned everything about how bacteria is cultured and how it is grown, how it multiplies. Okay, and after we multiply, after we get the culture by after incubating it, what do we need to do? We need to identify them. So we will also be able to isolate by various methods, right? Uh, Based on the nature of the microorganism. After we isolate, we also need to know how many cells are present. For example, when we have given a blood sample to a diagnosis, they tell us how so and so number of cells are present. How do they say that? They say RBC count, they'll say. They also say the lymphocytes, neutrophils, basophils, all these counts they'll be mentioning in our report. How do they do it? So there are certain methods in order to find the presence of number of cells. Our in our blood, RBCs are very small, right? They are very small, which is equivalent to a size of a microorganism. So when we can find the cells of RBC in our blood, or the white blood cells in our blood, and the eosinophils, basophils, all these when we can find in our blood, can't we find the bacterial presence in our blood? Right, we can also find. So we need to go with a similar method to that of the blood testing method, so that we can count number of cells present in our body or number of cells present in our sample, and therefore by that we can predict what is the quantity of infection, that what is the quantity of cells that are present in our body, and what could be the extent of our infection. So today's class objective will be to to learn about the measurement of bacterial cells as discussed just now, and various procedures to measure the bacterial cells. What are the various procedures? There are two types we'll be dealing in the next coming slides, and what are the advantages and disadvantages. So beginning with the cell measurement of cell numbers, there are two methods, two different categories of finding it. First is total count or direct cell count, which means we will be able to count directly the number of cells present. Whereas viable cells count or indirect method, that is by spread plate or pour plate method, where we can visually see the number of cells present. Okay, this is similar to that of isolation of pure cultures. That is similar to that one. So I won't be dealing with the viable cells count because it is similar to that of the previous one. That is spread plate and pour plate method. And membrane filter, that will come across in the total count or direct cell method count. Okay. In total count or direct cell count, there are four different methods. First is breed method or direct microscopy method. Understand the word breed method or direct microscope method, which means directly we will be observing through a microscope. Next is counting chamber method or hemocytometer method. Okay, and then third one is electronic counter method, and fourth one is proportional counter method. So we'll be dealing each now. First, breed method or direct microscopic method. What is actually done in this? In this, what is done is they take a glass slide and make a sphere. What is sphere? Wherever you put a bacterial suspension sample and it forms a round shape, that is called as sphere. So a known volume of microbial cell suspension is spread uniformly over a glass slide covering a specific area 1 square centimeter. So a glass slide, in that 1 square centimeter, just 1 square centimeter, it could be this much. 1 square centimeter in a round way, sample will be added. Okay. The smear is the round one, the sample one, which is round in shape, is called the smear. The smear is then fixed by heating. Okay, it is first heated, then stained by adding reagents. Okay, then examined under oil immersion lens, and then the cells are counted. How do we count? I'll tell you now. Customarily, cells in a few microscopic fields are counted because it is not possible to scan the entire area of smear. So it is not possible to scan the whole smear. So a particular area will be counted, and average of number of cells are present will be taken, and then will be counting number of cells, total number of cells present. The counting of total number of cells is determined by calculating the total number of microscopic fields per 1 square centimeter. So the glass slide that we put on, we have another cover slip that we put upon the sphere, okay, smear. On that microscopic cover slip, there are certain grids. It could be horizontal and vertical lines. So in those horizontal vertical lines, how many cells are present will be counting, and will make an average and then count the total number of cells present. So how do we calculate that? First, the total number of cells can be counted with the help of following calculations. That is, first, area of microscopic field. Since it is a round shape smear, so it will be pi r square. Circle area is pi r square, so pi r square. R is the oil immersion length. The lens that we see through has a diameter of 0.88 millimeter. 0.08 millimeter. That will be the very thin. It is like a small dot, or it is like a pinpoint, point which has the tip. It will be very tiny. So it could be it would be around 0.08 millimeter. So when we observe from 0.08 millimeter, number of cells that we see, okay, that we can multiply with the area of the sphere, right? First, I'm with one hole, I'm able to see two cells. Suppose if I'm able to see two cells, and I have a particular value that is 0.08 mm, and from that only two cells I'm able to see. So if I compare the whole pi r square, that is the area of the circle, and multiply it, won't I get the number of cells present? Right. So area of microscopic field is pi r square, and where r is oil immersion length, will be 0.08 mm in diameter. So therefore, area of microscopic field under oil immersion lens can be pi r square. Pi 3.14 into, okay, the r square value is 0.08. So pi multiplied with r square. I just said multiplying 0.08 with pi r square. That is not the case. Substituting the values of r square within pi r square. Okay, so 3.14 will be the pi value, and r square is 0.08 mm whole square. Okay, when I multiply this, I get 0.02 square millimeter. This was the average area of the field that I have taken. Now, area of smear 1 square centimeter is 100 square millimeter. 1 centimeter is 10 millimeter, but area of uh, sphere can be 10 into 10, so 100 millimeter. Okay, 1 centimeter has 10 millimeter, so area of 1 square centimeter can be 10 into 10, that is 100 millimeter. So the number of microscopic fields will be 100 divided by 0.02, because uh, the from the lens that we see, we get this value 0.02. So 100 divided by 0.02, that will be 5000. So number of cells present in 1 square centimeter is equal to average number of microbes per microscopic field. Average number of microbes per microscopic field. I have said there will be certain grid lines, right? So one microscopic field, whatever the number of cells are present, average number, we need to count for around 5 to 6 and take an average. It could be even, we can also take for 10 to 15 and take an average. From that average, we need to multiply with 5000, and then we get the number of cells present. Suppose let us take RBC itself. If in the grid box, I get around 5, in one grid box I get 8, in one grid box I get 12. So I take 15 readings and take an average of it. So average I can get around 15. Okay, let it be 15. So 15 into 5000 I'll multiply. Whatever I get, that will be the count of RBC. Similarly, in case of microorganisms, whatever cells are found, that will be multiplied in this way to get the number of bacterial cells present. So counting chambers method, that is by hemocytometry. You might have learned hemocytometry in anatomy of first year. In anatomy, we'll take the blood sample and check number of RBC present, right? This is the image of hemocytometer. Has a new bar, improved bright line. The lines you can see here, which are very tiny and it is hardly visible. The bright color in this, that those are the lines, or we can say the grid lines. Whatever grid lines you see here, that will be present here. Okay, and this is the new bar slide, and the two upward extensions are called as mounting support, where we put the slide or the cover glass on which the blood sample or the microbial suspension is added. Okay, and after that we observe under microscope for this one, sorry, for this one and this one. So these are the counting chambers which has a grid lines. Okay, and you can see, if we can magnify this more, we'll be able to see number of cells present. So suppose let us take this one, it has 2 cells, this one has 4 cells, this one has 2 cells, this one has 4 cells. So in this way we need to count it, beginning from here, 1 2 3 4 5, 1 2 3 4 5, 1 2 3 4 5, 1 2 3 4 5, 1 2 3 4 5. That means it is a 5 by 5 matrix, horizontal and vertical grid lines. So based on this, we will be able to calculate number of cells present by different calculations. What are the advantages of this? First is it is easy. Okay, it is just taking a slide, adding a sample and observing under a microscope. RBCs are very closely packed. For that reason, it would be very difficult. This is of RBC, not a bacterial cell. Okay, this image is of RBC. So, if a bacterial cell are present, there will be a dilution in that. So when it is dilute, we can easily see the microorganisms present in each grid line. Okay, and it is easy, inexpensive and quick, since microorganisms, the bacteria will be in very less concentration, so it is very quicker process and useful for counting both eukaryotes and prokaryotes. That means bacteria as well as uh, prokaryotes is for bacteria, whereas eukaryotes we say when it is a human RBC or animal RBC, anything, animals RBC, even we are social animals, right? So animals RBC from the blood, also plant cells we can count. Okay, and more easily counted if they are stained or if phase 1 contrast of fluorescence microscope is employed. What it means is, if we can stain the bacteria, staining the bacteria means it gives certain color. So upon observing in the microscope, rather than seeing a black and white image, if we stain, we can be able to see a colored image or colored structure, whatever it is. It could be red, it could be blue, it could be pink, anything. So based on that, we can identify a particular cell. That will help in both. And another one is phase contrast fluorescence microscope, that produces fluorescence when the light comes in contact with the bacterial cell. So based on that, we can also identify number of cells present in the grids. Okay. The disadvantages of this method are, it cannot distinguish between the dead and living cells. So all the cells will be present in that, dead cells will be present as well as living cells. So it cannot distinguish between them. And very small cells are usually missed, as some cells can come between the grid lines and we might miss them. Okay, calculated taking the count of number of bacteria per unit area of grid and multiplying it by a conversion factor depending on chamber value and sample dilution used. So the values will be varying based on the method that we follow, based on the grid box that we follow. Okay. The next is electronic counter method. Okay, electronic counter method. In this, we use electricity. So basically, electricity is used. For that reason, it is called electronic counter method. We'll see why it is called counter method. So basically, in this, microbial suspension flows through a small hole or orifice or capillary tube. So you can see this one. This contains a microbial suspension. Okay, this microbial suspension is passed through this one. Okay, this tube, and from this tube it will be so thin that only one cell can pass at a time. Only one cell can pass at a time. So what happens is, the sample is given here. One cell passes at a time, and there is a counting orifice. Orifice means small hole, very tiny hole where only one cell can fit. When that cell fits, there will be a detector on the other side. You can see that electronic detector here. So this detector, when the cell comes here, this detector immediately captures that and estimates the size of the cell. Okay, size as well as it counts every cell moving will be one count, right? So number of cells moving, it will be number of cells present. So the diameter of this tube is so microscopic that allows only one cell to pass at a time, right? One count thousands of can count thousands of cells in few seconds. So it means in very few seconds, thousands of cells can be counted from a microbiological suspension. Movement of microbe through orifice or capillary tube impacts electric current that flows through orifice. Okay. So movement of microbes, it causes electricity whenever the cell is coming. Suppose this is the orifice. Okay, when the cell is coming here, immediately electric current will pass. Okay, as a result it will be counted as 1. So the next cell come, it goes away, it will be counted as 2. The next cell come, it goes away, it will be counted as 3. This way this method follows. So advantages is quick and easy to use. As you have seen, thousands of cells can be counted in few seconds, right? And useful for large quantity, and microorganisms and blood cells can also be counted. What are the disadvantages of this? Cannot distinguish between living and dead cells. Counts even dust particles. Okay, dust particles can also be present in the suspension that we take. So even it counts dust particles. Suspension must be free from foreign particles. If it is free from foreign particles, then only it will be able to count the number of cells present, bacterial cells. Even if the dust particles or any other foreign particles are present, even that will be counted, and that will give an error. I mean, it will give a false result. And not for prokaryotes. Okay, this is not for bacterial cells. It could be used for any other things. Okay, if the size of the bacterial cells is a bit bigger, equivalent to RBC, then we can do that also. Moving on with membrane filters. What is membrane filter? By the word itself, you can understand membrane will act as a filter. So a diluted suspension of microorganism cells is filtered through special membrane. Okay, that membrane is a membrane filter that provides dark background for observing cells. Cells are retained on a filter. The risk is placed in a culture media in a petri plate, incubated ideal growth factors. You can see this image. This is the collector of the solution, and this is, it has opening at one end, and this is a membrane filter. When it is attached, the suspension will be added from the top. Okay, as a result, the solvent comes down and bacteria will retain on the membrane filter. This membrane filter is taken using a forceps. We cannot put our hand on it. Okay, it might be it is contaminated, so we might get contaminated, cause different infections. So we need to use forceps and place in a petri plate. So this petri plate, upon incubation for 24 hours, it shows the growth of microorganism, and then that can be seen under a microscope how many cells are present. Okay, and this membrane has every small part, every unit of the membrane is so small that only one cell can fit in that. So it is useful for counting bacteria. With certain dyes can distinguish living from dead cells. So certain dead cells does not react with certain reagents, and whereas the other cells, living cells, show reaction with the dead cells. As a result, it shows a color. Whereas the dead cells does not show any color. So there is a possibility of distinguishing dead cells and the living cells. So this was membrane filters. And as discussed, visible count method that are similar to that of 4-pet and split pet techniques. For that reason, I haven't dealt with it. It is similar that after we incubate, we will be able to see number of cells that are present after we stain them. Okay, and this is it with today's class. And the next class will be studying about the microscopes. What are phase contact microscope, electron microscope. We'll see in detail what is the principle and what is the procedure of that. Okay, and thank you for watching the video.